Okay, so yeah, I'm Stephen Voges. I'm a starting PhD at the NIN. I spent a lot of time there as a master's student, sometime as a research assistant, and now I'm finally starting my PhD, which is nice. And I work in the social brain lab, where we predominantly look at social behaviors, helping behaviors, social harm aversion, empathy-like behaviors, and I use the word empathy-like because it's difficult to speak to mice and rats and see whether they are really feeling each other's feelings. So we have to interpret this in any way we can. This paper got me really excited because it was taking MDMA research and putting it in one of the biggest journals in the world. And it had a social behavior, so I thought, this was incredible. So I kind of forced my lab to have a journal club about this as well. And I just wanted to point out one thing about this journal is that it has kind of one of the biggest names in neuroscience.
It has one of the biggest names in science, period.
Yeah, which is Edward Boyden. And when he was one of the people who was responsible for developing optogenetics in the form that it is used the most. So there were people who came before him that laid the groundwork, but he really kind of created modern optogenetics in the way that most neuroscientists use it now.
Yeah, I mean, I know Ed. I've been friends with Ed since he was a PhD student, and like, he was one of, I wouldn't say he is the main person. He was one of the main people, like him, Diceroth. There was also a guy in, I think, Wayne State in Detroit who like independently was developing, optogenetics, but he just didn't get any credit. Yeah, just want to give credit to all the people and not, Ed is an amazing scientist. He's a great guy. I still stay in touch with him, but like, he's not the main person. Like, there were several people who really made very important contributions, and Ed was definitely one of those people.
Yeah, and it's also just important when you see an author who carries that much weight to be quite critical about the work, because the reviewers aren't always going to be as critical about their work because they really want this author's next paper in their journals. So that doesn't mean that their work is flawed or anything like that, but it's also important to not get overexcited and go, oh, it's Ed Boyden's paper, so everything must be fine. And that's what I did when I first read the paper. And then when I read it with some more critical people, I learned that there are some issues, and there are also some really interesting things to be learned from it as well. So I think the main topic of this paper, in a way, at least from the perspective of psychedelic research, is MDMA. If you've ever taken MDMA, you kind of know what it feels like. You feel quite happy. You want to hug your friends all the time. So some studies show it increases empathy and pro-social behavior. What's really cool is that MAPS now has it on phase 3 for clinical testing for PTSD, which means that hopefully in the near future, it will actually be a therapeutic that you can just go to a doctor if you have very, very severe PTSD and get MDMA therapy. But also it can be kind of bad if it's used excessively because it messes with your serotonin system. And the main way it does that is, so these are serotonergic neurons that release serotonin. So these little blue triangles are serotonin. And they mainly do that through these little balls of serotonin called vesicles. These dock at the synapse and release the serotonin, which is then picked up by a serotonin receptor. And this can help with, this kind of leads to the feeling of happiness that we experience when taking MDMA. What's really interesting, and when I first had a scientific presentation on MDMA, is it kind of works like a reverse vacuum cleaner as well. So it hijacks the serotonin transporters here and prevents them from taking excess serotonin back into the neuron, and actually starts sucking serotonin out of the neuron. I think reverse vacuum cleaner was the way my professor at the time described it. You can think about it as a snowblower, for example. So it just makes a lot of serotonin available for the receptors, more than you'd normally experience. And it also does similar things with dopamine, but generally when talking about MDMA, people focus on the serotonin pathway. And releasing this much serotonin repeatedly can go wrong. And they've done some studies in monkeys where they gave them MDMA, I mean, twice. This is like the scary image. They gave MDMA twice a day for 4 days, I think. And in monkeys, it really showed a decrease in serotonin. And this, of course, directly afterwards was a lot less, but even 7 years, they didn't match controls. Of course, this was research funded to make MDMA look really scary.
Wait, is this from George Ricarte's lab? Is this that data?
I think so.
There was a study they did in monkeys. I think it was published in Science, where they claimed neurotoxicity after MDMA, but then it turned out they actually injected the monkeys with meth and not MDMA, so they had to retract the paper. And there were some suspicions of scientific misconduct associated with this.
I knew you would have a correction for me at some point, Rudy.
Well, I'm not sure this is that paper, but it could be that paper.
Okay.
This is monkey data, right?
Yeah.
Yeah. Okay. It's probably from them, but like we can figure this out later. Anyway, continue.
But yeah, I mean, it's important to realize that you're using up a lot of serotonin if you take MDMA, and that should be done with some kind of responsibility, I think. And that it will take your brain longer to kind of get back to normal levels than if you weren't kind of hijacking your serotonin system when you do it. And I think people have experienced this when you have that kind of like day after come down and you just want to lie in bed. So responsible use is something I would encourage. The next kind of important thing is the critical periods of plasticity. And I literally took the definition they used in the paper because I want to hold them to their own definition, which is a critical period is a developmental epoch during which the nervous system is expressly sensitive to specific environmental stimuli that are required for proper circuit organization and learning. And so they showed this in the 80s and 70s in cats. They would sew one eye shut and see how their visual cortex developed. And essentially the side of the brain that dealt with the shut eye was very underdeveloped, and the side of the brain that dealt with the open eye became overdeveloped. And you can imagine that, so there was a critical period for when their vision could develop properly, and by depriving that eye and all the neurons attached to it, they kind of ruined that critical period. And there wasn't really a chance to recover, although there's some research now showing that you can reopen critical periods and fix things. So that's what this paper is trying to do. I think the third important aspect, of course, is oxytocin. And this is what I find most fascinating about this paper. So it's a neuropeptide that's really important for bonding. So classically in prairie voles, which are kind of small rodents, they have kind of much more oxytocin receptors. So all these dark areas are oxytocin receptors in their brain, compared to meadow voles, which are almost identical animals. And what happens is prairie voles are monogamous and meadow voles are non-monogamous. Interestingly, prairie voles are still promiscuous, so they still cheat on their partner, but they really have one partner that they mate with for life. And this is due to the oxytocin receptors in their brain. It also is important for reproduction, childbirth. When they induce labor in women, they generally give artificial oxytocin injection, which I've heard isn't very nice. But oxytocin starts the process of childbirth. So it kind of has multiple roles, both as a neuropeptide and as a hormone. And there's some recent research that shows that it's important for emotion recognition in mice. So it's definitely kind of an emotional neuropeptide. It has to do with bonding, reproduction, childbirth. And it's really what the key of this paper is. And actually, this paper was inspired by a previous paper of theirs, where they found that oxytocin was responsible for social place preference. And so the main behavioral paradigm that they use in this paper is SPP, as they call it, or social place preference. And they use this as a proxy for social reward learning. So pretty much the mice are taken from, they're kept in a home cage for 24 hours or more. I don't really understand what they're trying to say here, that maybe that they're kept for longer than 24 hours with other mice. And then at the beginning of the testing, they're first allowed to explore this box. And the box has two sides, which they have here as purple and green. And they can just walk around it. And here they're measuring essentially what side they have a preference for and how much time they spend in either side of this box during the beginning. After which they either expose them in for 24 hours to one side with other mice, so making that side associated with social context, or they expose that animal to the isolated side for 24 hours. And then after that, they switch it. So it's kind of randomized, but they either do social conditioning first or isolate conditioning first. And then next they do whichever one they haven't done yet. And then afterwards, both sides are opened again and they measure again which side they have a preference for. And so the idea is, if they have a social preference, then they'll stay more in the green side during the post-conditioning trial. But if they have a non-social preference or they don't care about the social preference, they'll stay in the pink side. And what they say is that this is a form of social reward learning.
So I would say actually that, like, if they don't have a preference, that means that they just don't have a preference. And if they prefer the purple side, then they're actually preferring being alone.
Yeah, so there's no preference. And there's also, preferring being alone, which some people actually do. And it's not inconceivable that some animals also prefer to be alone. And that might be based on hierarchy. If you're bullied a lot, you might not want to hang out with all the other mice. And mice can be really rough on each other. I don't know if your mice fought a lot, Rudy. But I definitely have male mice that like to harass each other and bite each other's ears and do nasty things to each other. So it's not like they're all happy. Rats would probably be a better model here because they're a lot nicer to each other, and their play is intense, but they don't seem to get as upset with each other. But this is kind of the behavioral paradigm that the entire paper is based around. And so what they wanted to look at is whether this paradigm, if you do this over different ages, so from post-weaning, so after they stop breastfeeding essentially from their mothers, until adulthood. So you have kind of post-weaning, sexual maturity, and then early adulthood. And these are defined by days since birth. So on this next slide, which is a lot of graphs, P21, this is the post-weaning. I think P49 or P42 is when they start reaching sexual maturity. And then adulthood becomes quite complicated. I would say around P84, that's like early adulthood. But often people like to argue that mice or rats are adults earlier than they are so that they can start their studies faster. But I think there's a lot more development in mice and rats than is given credit for after the sexual maturity. So they did this test, and here.
Hey, I'm sorry, I would like to interrupt something. So Hesha was asking some questions in the chat.
Oh,
And
I can't see the chat.
yeah, it's okay. It's okay. No. So one thing that I was unclear about, maybe I just spaced out for the time you were saying that, is the conditioning done in the home cage or in another cage?
So it's not. I don't think it's done in the home cage.
Okay,
no.
yeah, it's usually not done in the home cage. Yeah, I kind of assumed that it was not done in the home cage, but I just want to clarify because.
But considering it's a 24-hour one, there's definitely some kind of food and water available for them, because otherwise they would.
Well, yeah, I mean in the home cage, yeah, but no. My question was just: is the conditioning environment in the home cage, or is it a different cage, a different box?
I don't believe it's the home cage. I'd have to kind of
here. I can check for you. You can just continue. I don't want to hold your presentation.
Because I generally assume that these conditioning trials are not done in a home cage.
Yeah, me too. I mean, that's a general assumption in the field. Okay, sorry. I'm going to look this up. You can just continue.
Yeah. So 900 is kind of, I guess, the midpoint, but it goes. Their graphs are a bit confusing, but 900 would be kind of an indifferent score. So they spend as much time on either side. Towards the 1500, you have the social, and towards the 300, you have the isolate. So what you see is at the beginning of the trial, there's quite a broad difference in what they care about, but then they haven't been conditioned to any side yet, and after they've been conditioned and they know that the social side is the social side, they begin to lean up towards this social side. Can everyone see my mouse? Is that showing or no? Okay. And so they do this test over time, and what begins to happen is there's if you look at the average of the time spent, this begins to flatten out over time. And by P63 already, it's no longer significantly different statistically.
Sorry, P is the P number is the age, right?
Yeah, so it's number of days since birth, essentially.
Yeah, P stands for postnatal.
Yeah. And so it flattens out and becomes much more neutral. But what's interesting is up until about P49, the data looks really nice. Before, they're all kind of clustered together, and afterwards they do mostly lead in one direction, although there seems to be, even in the beginning, lots of animals that look indifferent or even go the other way, or some animals go the other way. It's a bit difficult to see with these lines, but here there's some kind of almost flat lines where they don't seem to change their choice from what they liked before. So this is kind of where the questions begin to come in on if they're measuring what they think they're measuring. And at P56 onwards, you see that the data gets really scattered, especially the baseline data. And I'm a bit curious whether this was fully normally distributed, because when you do the t-tests, the statistical tests that they use, the data should be normally distributed. And looking at it by eye, it's difficult to tell, but it looks like it might not be, which makes me question whether they were using the appropriate tests for their data.
Why do you say it might not be normally distributed? Like, why do you say it looks like it might not be normally distributed? Just because there's not a lot of stuff in the middle?
Yeah, I mean, that's what normal, I mean, that's kind of one of the important parts of a normal distribution. You do have this kind of middle cluster, right? That's
yeah. Well, did they say in the methods whether they tested for normality?
they said they tested for normality, but I also think they used the t-test on every single thing in the entire paper.
Okay, that can't be right. Like using the t-test that much can't be right. But you know,
yeah.
this is so. As a bachelor's student, I worked in the same lab where the senior author of this paper did her postdoc, and like there was quite a bit of inappropriate use of statistical tests in that lab.
Is that Malenka lab?
Or? Malenka, yeah.
Yeah.
I mean, I didn't realize it at the time because I was young and I didn't know that much statistics yet. But then later I was like, whoa, like people were just, you know, really playing fast and loose with the statistics just to get significant results.
Yeah. And so they create this preference score. And I think the idea here is that 1 would be no preference, and anything above 1 would be a social preference. Anything below 1 would be an isolate preference. And you can see over time that it decreases to more of a neutral preference. And the subtraction score is similar, except they make a score out of the difference between the social versus isolated. So I think it's a ratio versus a subtraction, and it pretty much gives the same data, except here it even goes under the 0 mark. So 0 would again be neutral here, and below it would be isolate preference. What's interesting is it does seem to stay over, not significantly, not statistically interesting, but it does seem to stay over the 1 mark and 0 mark most of the time, even at the older ages, which I think should still be taken into account. So what they claim here is that there is a critical period for social reward learning, which closes here at P63. And my question really is that a critical period? Are they saying that these animals no longer can do social reward learning? Is the test demonstrating that? And are the conclusions they draw after this actually showing that they're reopening a critical period? I think that's a nice point of discussion, but maybe we go to that after we've looked at some of the rest of the data.
Well, so I will already say my personal editorial comments as the answer to your first 2 questions is no, and like the third one we can get back to later. But like, yeah, CPP is like, it's got a lot of problems. I mean, I think it's, you know, it's simple, which is why it's simple, it's fast, which is why so many labs use it. And you know, it does tell you something like you can say, okay, well, you know, this is if the animal get conditions to the side where, you know, to one side where you gave a specific stimulus, then like I think on the basis of that, you can say that stimulus was an appetitive stimulus. And you can draw some inferences about reward. But like, I really don't think, you know, as somebody who I actually started in neuroscience doing CPP experiments, and like as somebody who's done these experiments, like I really think you can't interpret it any more than that, because like, yeah, it's just, it's, you know, it's a noisy paradigm. Like you get like really broadly distributed data, which is sometimes not normal, and like, you know, that kind of makes it a little bit more difficult to like draw really strong conclusions about reward just based on this behavioral paradigm. But I think it's a decent place to start. I was hoping the paper would contain more than CPP, so that was kind of I was a bit disappointed, but anyway,
so the entire paper is based on CPP or SPP in this case, but they do some like cocaine CPP to see if this is maintained. But yeah, I think a task where kind of the animals had a choice between an area that they knew would be social or an area that would give them like a food reward, so something else that's valence, would have been a more interesting paradigm, but also more complicated. But yeah, so after this, they wanted to look at what happens to nucleus accumbens neurons essentially over this time. And they'd already seen that oxytocin was important for this paradigm in a previous paper, and they wanted to see how this changed over the age course. And what they see is that young animals have this long-term depression, which is essentially you give a specific type of stimulation to neurons, and they kind of say, well, that's enough stimulation, and they stop responding as much. And it's an important part of plasticity, so being able to learn correctly, because if you just accumulate all the information that's given to you, you're not able to process information properly. This is kind of a hand-wavy, basic explanation of what LTD does. But what's important is that it plays a really important role in plasticity. So what they argue here is that there's a decrease in plasticity in response to oxytocin, which they don't see when they use a serotonin agonist. So they think maybe oxytocin, these changes in oxytocin are responsible for what they see in the behavior. Okay. And this, I think, is actually likely, and they do demonstrate this, I think, pretty well later on in the paper.
So hold on, before you go to the next figure, I have some comments about this one. So yeah, basically LTD is a form of plasticity where like glutamatergic, so the glutamate is the main excitatory neurotransmitter in the brain, and so like the brain needs it to like to stimulate activity. And so here basically what's happening with LTD is a form of plasticity, and so you can see the in the oxytocin condition in P40, like after they stimulate, give their stimulation protocol, then the strength of the synapse goes down. And so that's what you're seeing on the y-axis. So basically 1.0 is like the normalized, like normal strength of the synapse, and anything significantly below that means the synapse got weaker.
Yeah. Thank you, Rudy.
And so, yeah, in A, you can actually see like, 1 and then 2 is where the LTD happened, I think. Yeah.
Yeah, so and this is also where this measurement is taken from. So minutes 50 to 55. And yeah, so this is what they're going to base their argument on a little bit. But they also wanted to check whether the older animals still exhibited normal conditioned place preference. So they did cocaine CPP. And young animals and old animals show similar behaviors in response to cocaine. So they prefer the side where they get the cocaine over the side where they get just saline. So it's the same paradigm, you just replace social and isolate with cocaine and saline. But again, interestingly, the older animals have this weird distribution again, which I can't really make sense of, but it seems like they're more likely to pick a side and stick to a side than the younger animals, which seem to be moving around more and seem to be more indifferent in the beginning. Any comments, Rudy?
Sorry, I was kind of spaced out for this figure, but yeah, I mean, this is, I think, a really good control, because like you want to show that it's specific to the social reward and not general to drug reward. It would have been nice. I mean, it would be nice if they had shown like other types, like tested more different reward conditions. But because it's oxytocin, I think you can be pretty confident that like it's gonna be social. But like it would still have been nice if they had tested more like non-social reward conditions.
Yeah, because I also think like cocaine is a pretty like big condition to use as a comparison, and something a bit more subtle might have been nice to see if it yeah, if other things work, because I think social stimuli are quite subtle in many ways, especially for older animals. So it would have been nice if they didn't take a sledgehammer approach to their controls.
Well, I mean, I think this is a decent control. I just wish they would have had more controls in addition to this one.
Yeah, for sure. So this one is busy, but I couldn't really figure out a nice way to split this over slides. So what they see is, so here they start with the MDMA, and first they look again in the slices and they look at this LTD. And what they see is in the older animals, if you essentially add MDMA to these slices, there is LTD, and which is this blue line here. You see it's going down, so it restores the response that they saw before. But if you block oxytocin receptors with an oxytocin receptor agonist, which essentially prevents the MDMA from what they're assuming binding to the oxytocin receptors, or something in the circuit of the oxytocin receptors, this doesn't happen. So here they're showing that MDMA is reinstating this LTD, and that it's oxytocin related, which is quite nice, which is really showing that MDMA does affect neurons with oxytocin receptors, which is kind of adding a lot to the field, I think. So they, of course, wanted to see behaviorally, and this is their pièce de résistance, whether they could reinstate this social place preference with MDMA pre-treatment. So they use a very similar paradigm as before, except 2 days before the testing, they get either saline or MDMA, and this was 10 milligrams per kilogram. So if you were to,
so that's a lot in human.
So that if you were to think about a human being, that's a lot. But I mean, mice do metabolize things different than humans. But it's not exactly a low dose of MDMA. It's a pretty intense dose, even for mice. And
sorry, but so they're giving them MDMA like before the whole conditioning, not during.
Not during, no.
Okay.
So the idea is that the MDMA is having a therapeutic effect rather than a direct effect on the behavior. So it's not that the animals are under the influence of MDMA during the experiment. It's rather that the MDMA has done something to the plasticity in nucleus accumbens. Well, they show later that it's possibly in the nucleus accumbens, but this is what they're going for, is that it's not an effect of the high rather than an actual change in the brain. And yeah, they show here that in the older animals, if after you give them MDMA, there is an increase again in this social place preference. They prefer the social side again. Again, they use this, what's it called? This preference score to exhibit it, and you actually see a higher level of preference after MDMA than you even saw in the animals from the previous graphs that were younger, that were showing high social place preference. And they do this as well with cocaine, I guess to see how dopamine plays a role or how another drug may play a role, and they don't see that cocaine can increase this social place preference. But I'm not entirely sure if cocaine is if that's a great comparison, but I guess it's interesting that you can't just give any drug and it will increase social place preference. It's specific to MDMA, or it's at least not specific to cocaine.
Yeah, I mean, okay, I think this explains a little bit more to me why they chose cocaine as their one control, because like I think this is a pretty like nice, elegant comparison, like same design, just a different drug. So and I think other non-social rewards, it may have been more difficult to do just the same design of the CPP experiment.
Yeah. And also looking at the animals that had the MDMA pretreatment, they show that this LTD, this long-term depression, is restored in the older animals who had the MDMA pretreatment. So that's actually quite interesting, I think. So it's showing that there's actually a change in plasticity happening when you with the oxytocinergic system in the brain due to MDMA. And for me, this is kind of the most interesting part of the paper, that MDMA can really change the plasticity in this oxytocinergic system. And I could take or leave the rest of the paper, actually. I don't know how everyone else feels about the other parts, but I think that this is actually kind of the biggest highlight for me in terms of what they found in the paper so far.
So to see if I understood, basically they're showing that the treatment with MDMA kind of brings back the sensitivity to oxytocin of old mice. It kind of makes it more similar to the one of younger mice, if I understand correctly, in terms of this LTD. Or is that not what this is showing?
It's showing that it restores the behavior of oxytocin neurons.
No, these are Cummins neurons. Are they? They're recording here in Cummins medium spine neurons, right?
Oh yeah, shit.
Yes. So I don't see how you can make any relation to oxytocin with these figures. Isn't it just specific? It's promoting LTD.
I think that was in her paper, like Dolen's paper, where she was her big paper from her postdoc in Rob Malenko's lab. So I think that experiment is actually already in previous work that she did.
Okay.
Yeah, let me look at my things. But she didn't do MDMA in that experiment, Rudy.
No. But I mean, I think if I understood Hesho's question correctly, she said that in this figure they did not show that the LTD is dependent on oxytocin, and I'm pretty sure that they did show that in a previous paper.
Yeah, they definitely did show that in the previous.
Wasn't it also the previous figure, like that you had some slide ago? Wasn't that one about showing that oxytocin does LTD in young mice and not in old ones? You go back, was it one slide or two? Yeah, isn't that the top one here? Showing that oxytocin does LTD in young mice, but not in old mice, right?
Yeah.
Yeah, so it's comparative effect, and then of course, yeah, there's the serotonergic receptor. Okay, yeah.
Well, this is what I didn't understand. Why did they compare this with like a 5-HT1B agonist, like kind of?
So that's serotonin.
Yeah,
but why choose that comparison?
Sometimes that's the only agonist they have in the fridge. I don't know.
Yeah. So I have some. Stephen, first you answer, and I will give my comments.
Well, I think that if you're building up a paper about MDMA, you definitely want to also be looking at serotonin over your paper. So I think that showing that this isn't happening with the serotonin agonist is important. So I don't know what Rudy's answer is, but
I agree completely with Stephen, with what you said, Stephen, and I will extend that by saying that that 5-HT1B receptors are highly expressed in the nucleus in the nucleus accumbens, and I think throughout the striatum, and they have previously by other labs been shown to be important for drug reward and other behaviors, other reward-related behaviors.
Right. Thank you, guys. That's very interesting. And also, again, I guess the point here is that oxytocin has like an age-dependent kind of effect, whereas the 5-HT1B agonist is the same for it's independent of the age.
Yeah. So that's also a good point that you make, Marco. That's also like a nice thing that like bolsters their interpretations of their data.
Yeah. What I did find curious was kind of how they show their data. So in this one, they leave the older animal under the younger animal, and in this one they put the older animal over the younger animal. Which
Interesting, yeah.
I don't know if it makes it look any different if you were to switch it around, but it's an interesting way to depict two figures that have like similar information, right? So. I mean, that's, I think, nitpicking a little bit. So the next question they wanted to really describe is this, because they've shown all this slice work in the nucleus accumbens, is this actually in the nucleus accumbens? Because before they were doing kind of just general injections into the belly of the animals, I'm pretty sure, intraperitoneal. And so they put a cannula in the nucleus accumbens so they could directly infuse drugs into the nucleus accumbens. So it's just a little tube that you can put in there, glued to the skull, and then you can infuse whatever you like into the brain, directly into the region that you're targeting. It can spread out of the area, but the nucleus accumbens kind of big enough that you're not going to worry about that too much. And so they did a comparison of if you just infuse directly MDMA into the nucleus accumbens, or if you infuse MDMA and an oxytocin receptor agonist. So this would reduce the ability to bind to oxytocin receptors. And what they show is if you just give MDMA, you get this social place preference. They're more social. But if you add the oxytocin receptor agonist to the mix, they don't show this social place preference. So they don't increase their social preference, even though they are still more social than preferring isolated. So that kind of shows, okay, there's a connection here between MDMA and these oxytocin receptors. So their next question was, what if we directly stimulate oxytocin, I think oxytocin receptors, no, oxytocinergic neurons in the nucleus accumbens. Oh no, yeah. And use optogenetics, so you use a light to essentially switch on these neurons specifically during, it's not really clear when they're actually stimulating it.
I think there must be doing it. I mean, I haven't looked at the methods, but I mean, I would just do the stimulation in the home cage, because like just the same as, you know, like when they give the MDMA or the cocaine.
Yeah, it would have been nice if they put an arrow here to when the stimulation is happening. And I'm sure if we look in the supplementary figures, we'll find a bit more about how they stimulated it. But Kronos is a really kind of fast optogenetics. So you can do very precise stimulations.
Yeah, and the neurons will follow it, Faith. If the neurons are capable of firing at really high frequencies, like they will be able to follow the frequencies that you want to fire them at, because Kronos has extremely fast kinetics.
Yeah, so hence the name Kronos, because you can lock your neurons into your optogenetic, right? And by directly stimulating these oxytocin-dependent neurons, they were able to, again, recapitulate the behavior, and they just have a wild type control again, just to show that this doesn't happen in the control animals. So the picture they've kind of painted here is that, you know, MDMA reinstates the social place preference, that oxytocin receptors are definitely involved, and that if you directly stimulate oxytocin neurons in the nucleus accumbens, that you can also reinstate the social place preference. I think there's still a bit of a connection missing between some of the stuff, but I mean, you can't cover every single connection that there is in the situation. But what they're really illustrating is that there's a relationship between MDMA and the oxytocin neurons. And so they...
Yeah, so I would like to go back. I mean, again, did you explain, like, in detail, like, the interpretation of the probability distributions of the postsynaptic potential of the synaptic potentials in figures I through M?
No, I didn't.
Okay. So basically here, I think this is actually quite important for making one of the connections between the MDMA and the oxytocin. So basically, what they show here is that when they give in the oxytocin receptor agonist, so basically, so in I through K, I mean, sorry, in the left part of the figure, so the black trace in I and then J and K, I'm sorry, like they're showing the distributions of the synaptic responses. And what you can see is that the distributions are different between ACSF and here, and so in J, they're showing like the frequency. So basically interval, which is one way to how frequent are and these are, I think, probably miniature postsynaptic currents, which means they're just like spontaneous release from the presynaptic terminals of the neurotransmitter, and they're not actually mediated by action potential firing. And so this is one way that people can get an idea of, like, you know, how strong individual synapses are, because the idea is that these spontaneous neurotransmitter release events, like, are only going to occur like one or a few synapses at a time, because they're not triggered by action potentials. So it's just kind of like and a small minority of cases, the synaptic release machinery will spontaneously fuse with the membrane and cause neurotransmitter release. And so therefore, what they show in K, well, in J and K, is that when they give the oxytocin receptor antagonist, the frequency of the synaptic responses increases. When they give the OTR antagonist, it blocks the decrease in the frequency of spontaneous synaptic events that occurs after a social reward learning. And however, when you look at the amplitude, the distribution of the amplitudes of the responses, you don't see any effect. And so what you can learn from this is that, so basically, if you think about maybe, Stephen, it's good to go back to your figure with the axon terminals in it, just so people have a visual aid.
I did something, 1 second. Okay. This one or the original one?
The other one is fine too. Hold on. Maybe can you go to the other one?
And I've screwed it. Sorry. So this is their working model.
Okay, actually, why don't you go and explain this working model, and then we'll return to the synaptic recordings, and then I'll kind of explain the rest. I think it'll be easier for like the non-neuroscientists to understand if I do it that way.
Okay. So they present, it would have been nice if they just did this as one kind of giant working model, but they kind of separate each part of their paper into a different working model. But the basics of it is essentially they're showing what is going on in the nucleus accumbens and how, you know, oxytocin neurons, serotonin neurons, and glutamate neurons are all getting together and acting on these nucleus accumbens medium spiny neurons. And so here the oxytocin neuron activates the oxytocin receptors in the 5-HT neuron, which in turn activates this, this serotonin receptor on the glutamate neuron, and ends up, remember, like Rudy said, glutamatergic transmission is kind of like the main excitatory transmission in the brain. This in turn acts on the nucleus accumbens medium spiny neurons. And what they argue is that there's a decrease in oxytocin receptors on the serotonergic neurons in the adults, which I'm not sure if I'm fully there, but maybe the ePhys shows that.
I mean, maybe there's something in the supplementary that I'm forgetting about, but like, I don't think they provided any direct evidence that there's less OTR in adults, or maybe it was in a previous paper. I don't remember, but in this paper, I don't remember any direct evidence.
In the previous paper, I don't think they look at older animals is the issue. So I'm a bit confused as to this part of it. So my recommendation is that we just don't look at this for right now, and we look at their MDMA reinstatement hypothesis, which I think is quite nice, and it shows kind of a recursive system that's happening here, so that you have so with the MDMA you're activating these serotonin receptors on the oxytocin neurons, which in turn are activating these oxytonergic receptors. I'm guessing that this would cause some kind of plasticity, so you would increase the oxytocin receptors if we're going to go back to this idea, which would increase the release of serotonin to the glutamatergic neuron as well. And finally act on the medium spiny neurons. And their optogenetic model is very similar, except instead of having the MDMA increase the serotonin to the serotonin receptor on the oxytonergic neuron, they're just directly stimulating the oxytonergic neuron in this situation. Yeah, I'm still not really sure how to make sense of the decrease in oxytocin receptors. This might be why this was far in the supplementaries and not one of their main figures, but I think that this is an interesting model. And then maybe you can explain the ePhys a bit more now, or?
Yeah. Well, it's just helpful to have these diagrams. So basically, I would like you all to focus on the blue axon terminal and the purple postsynaptic the purple postsynapse, because that's the synapse where they did their plasticity measurements. So, and this actually figure gives another reason why they use the 5-HTR as a control, because of its effects on glutamatergic inputs to the striatum. So, but yeah, not only does it affect the glutamatergic release, but the prediction would be that it would affect glutamate in the same way as oxytocin, because oxytocin, according to their model, is directly upstream of the 5-HT1B receptor. Okay, anyway, back to the synaptic stuff. So basically in figures I, J, and K, they're looking at the presynapse, so the purple, the blue one, right? Can you go back to the slide? And so what they're basically showing is, so if there's a higher frequency, that means there are more spontaneous release events from the blue terminal. And so then that basically means that something changed in the blue terminal to increase so there was some change to the presynaptic release machinery or some other signaling cascade that governs neurotransmitter release that increased this probability that the synaptic release machinery will spontaneously fuse with the plasma membrane and release neurotransmitters. So basically so they're showing that the plasticity, that they're providing evidence. Sorry, this is not 100% certain evidence, but they're providing good evidence that the location where the plasticity occurs is in the presynaptic terminal. Because if you had plasticity in the postsynaptic terminal, the main way that happens is by more glutamate receptors getting put in to the postsynaptic part of the terminal. So you can see these 3 glutamate receptors like that are on the postsynaptic density of the purple medium spiny neuron. And so therefore, if the plasticity is happening postsynaptically, you get more or less receptors. And therefore, you should see a difference in the distribution of the amplitude of the synaptic responses, but not the frequency. And so based on that data, it's evidence, it's not 100% solid, but it's evidence, it's good evidence that the changes are occurring presynaptically to the nucleus accumbens medium spiny neuron. Was that clear to everybody? Does anybody have questions about this?
It makes it a bit more clear also to me why they were using the 1B the 1B manipulation, right? Because the 1B manipulation is the part that all of these things kind of have in common.
Right.
So if you do that,
Yeah, I feel like they could have used this diagram, maybe not each working model, but a basic version of this diagram earlier on in the paper. And this was like hidden all the way in the back, which I found, maybe I could have put it a bit earlier on in the journal club, but I found it strange that I had to like go to the second to last figure, I think, to find this.
Yeah, I mean, I think it would have really helped just to have like one panel with just with the setup of the different, of these synapses, without the model, just showing here are the synapses, here are how they relate to each other, and like here are the different receptors we're targeting. I agree that would have been extremely helpful. And I'm working on a manuscript right now too, so I will definitely keep this in mind for my manuscript.
Well, I know, like with Nature publishing, you kind of, especially with a letter like this, which is just a really short journal, you hardly get to put any figures in your main body, right? You almost everything ends up in the supplementaries. So I understand maybe why they were trying to like fit in as much data as they could, and then have some nice things to help understand later in their paper.
Yeah, that's also a valid point.
There's also a lot of controls in the like basic controls for like the expression of the optogenetic and stuff like that in the supplementaries. I don't really think people are interested in that, so I didn't add it to this journal club. They also show that so most of this behaviors you've seen, or all the behaviors you've seen so far, is male mice. They show a similar profile in female mice, but then do the rest of the study in male mice. So this is kind of the point where they say they think that MDMA works as an effective therapeutic for PTSD because it reopens a critical period for social reward learning, which they actually say at the end of the paper. And for me, that feels like a big stretch. They definitely show a really important role of oxytocin with MDMA and increasing social place preference. And if we think about kind of how people say that they feel like they're bonding more when they take MDMA, and that oxytocin is considered the bonding neuropeptide, it opens up to a lot of extra ideas for research, for sure. But I'm still skeptical about the social reward critical period hypothesis that they're really throwing out as their main argument. And I don't think it's a critical period. I don't think it qualifies as a critical period, because they're not showing that if you do social place preference with a young animal and then do social place preference with the animal later in their life, that it's maintained because you've opened up this, you've enacted this circuit at an early time. They're just showing that this circuit changes over age, which is also not surprising, right? But that doesn't necessarily mean it's a critical period. I would really like anyone's input on this, because I find it the most frustrating argument of the paper.
This is speculation. Like, I don't think, like, I mean, of course it's nature, so they have to speculate like that to like get people's attention. But like, yeah, I mean, I think you can't really, that's a huge stretch. And, you know, they need more experience they need way more experiments to provide evidence that this is actually the mechanism of how, you know, MDMA assisted psychotherapy, you know, cures PTSD.
Yeah, but certainly, I mean, I think this paper is clearly a very good precursor for many more studies to come to follow through from this. So sometimes they don't want to shove it all into one.
Yeah, and I think it also opens it up for, like, you know, people are scared to do psychedelic research, right? Because it's messy, it's complicated, it's people judge you for it, whatever reason that goes around it. And having a big paper like this opens up the field, and I would be interested in using MDMA in my research if I could ever convince my boss to allow it, you know?
Absolutely. I'm slowly throwing taps at my supervisor. Let's see how it goes. It's a bit more strict here in St. Andrews, but fingers crossed.
Yeah.
Yeah, I mean, it's an uphill battle to like try to convince people who like aren't interested in psychedelics research to like get into it. Like, I have a colleague at the Institute who's like one of the star postdocs at the Institute, and like, you know, I mean, she's gonna have a really successful career. She's working. Oh, Zing, you know Zing, Stephen, right?
Who?
Zing Chen from Peter's lab.
Yeah.
Yeah. So anyway, like, she actually wanted to do like look at the psychedelic effects on the visual system in monkeys, and Peter was like, no, sorry, he is way too conservative. So
yeah, but I think as far as conservative goes, Peter is one of the most conservative PIs at the Institute.
Yeah, I mean, he's very old school. You know, he's a dinosaur. He should be replaced by somebody who can provide leadership that's more appropriate for 2020. I mean, he has good intentions. Like, he has good intentions, and like, you know, I think he genuinely cares about the well-being of the people in the Institute and the success of other labs in the Institute. But like, just the mentality is so backwards.
I think he has a very strong focus, right? And that's curing blindness, and anything else.
Well, his lab does a ton of other work on the visual system. Like, they do great world-class work. Like, he's a big lab. They do lots of different stuff. Like, I think psychedelics is totally like could be within the scope of his lab. Like, if he wanted it to be. Like, it's not a big stretch compared to the other stuff his lab is doing. His lab, I think, is one of the labs in the world that is actually best positioned to get into psychedelics research.
Yeah. But there, you know how the stigma is, you know, a lot of people, especially that do like vision, and especially that do like animal research, they see themselves as like very serious scientists with a reputation, and so they feel like they could be, you know, like their reputation would be damaged in some way by getting into like research about psychedelics. But you know, hopefully that will change a bit.
Yeah, I also think it's the challenges in getting this research approved as well. I think if you say this to a professor, especially in Europe where everything's still a bit more controlled, which generally I appreciate that we have a lot of like good ethics in Europe, but it's also a lot of extra work. But like, if you say like, oh yeah, I want to do an MDMA experiment, they might just not even want to think about the cost of going through all the approvals and getting everything set up so that they could actually just even store MDMA in a room with permission from the government. So, I mean, like, I think cocaine is more accepted already, but I think there's still a lot of work that goes into the regulatory approval of being able to use cocaine, right?
Yeah, there is.
Yeah.
But I mean, I don't think there's more. I think the work for MDMA and cocaine is probably the same. Like, I don't think there's a big difference in the amount of work. Like, in terms, I mean, you know, but I mean, for cocaine, like, almost all the research is like, you know, aimed at, you know, showing how cocaine is bad for you. And it is like one of the, in terms of like negative health effects, it's definitely like one of the worst drugs. And so of course, like, but like for this like the opposite. They want to show how an illegal recreational drug has therapeutic effects. And so that's definitely like not within the dominant paradigm of recreational drug research. And so I think this stigma definitely makes it harder.
But I think that also stems from a lot of the funding for drug research being like, you've got to show its harmful effects. So when you apply for the funding, say, I want to show how bad this is. And I think that needs to change in order for there to be more effective research into the nice things about psychedelics.
Yeah, absolutely. I mean, I think it's so interesting. Maybe let's finish the paper and also stop then the recording. I don't know if you have any other slides or anything as to.
No, this was the last. I mean, I just wanted to say what I've been saying the whole time. Whether it's a critical period, whether the test is a valid test of reward learning, and I'm still a bit skeptical about this dose. I mean, I did some quick math. It would be 700 milligrams of MDMA, or if you take an average pill has 100 to 150 milligrams, 4 to 7 pills. Which I think the scientific term for what those mice were is thunderfucked.
I think they usually do that, right? They want the full extent of the drug working. Like even with MDMA in the treatment of PTSD, they give the patients like quite a large dose of MDMA.
Yeah, per session they give between 80 and 120.
Yeah, they do it. Yeah, 700.
Mice and people, and like what you were saying, Rudy, about the actual pharmacokinetics of mice. It's
yeah.
I mean, it's tough. It's because of the big differences in pharmacodynamics and pharmacokinetics between species. It's really tough to know like what your dose is actually analogous to.
Yeah.
So I mean, I assume like you looked at the literature to get this 4 to 7 pills figure, right, Stephen?
Yeah, it's based on a pill being the average pill that the police pick up and measure is 100 to 150.
No, but I mean in terms of like the translation between mice and people, like that's.
I couldn't find something that really, and this is the issue as well. It's unclear, like how much mice are being affected by the 10 milligrams per kg. I know for like neurotoxicity studies, they give 20 milligrams per kg. So I don't know if it's comparable. But I'm still I still think that it's a high dose.
But you can also test for that.
I definitely think I would assume that it's a high dose, given what we know about the pharmacokinetics, and then like, yeah, I assume it's a high dose, and then like, hopefully like there's, hopefully there's something in the literature, like some model that will enable a lower dose that will enable them to give a lower dose that's like closer to the therapeutic dose.
Yes, they can also test for certain effects like head twitching, right? So which is like the equivalent to gurning, which humans have with MDMA. So they can kind of test to see these other kind of behavioral effects it can have on the animal. And I'm guessing they would have scrutinized that effect as well.
Yeah.
Yeah, so I'm not sure that head twitching is actually the equivalent of gurning, because it's supposed to be a psychedelic specific effect, and you get gurning with stimulants too.
Yeah.
So, but I think head twitch also is a 5-HT2A mediated behavior. So for like these particular experiments, it might not be good, but I completely agree with your critique that they should have measured more behaviors just to get an idea of how this dose of MDMA affect them. Like locomotor activity, for sure, I would have measured, and maybe their setup prevented them from doing that. And then I don't know. I probably would have measured some other simple behaviors too.
Well, I think they got the MDMA in the home cage, right? So I think that.
Oh, right, yeah, okay, that makes things more difficult.
so they were group housed and in a home cage. So I think keeping track of which I don't know if all the animals underwent the treatment and then they were mixed and matched together, or if they had special compatriots. I didn't fully check every line of the methods, but yeah, recording the homepage is always a pain in the ass. So I understand why there's less info, but it wouldn't. And they said they tested it empirically, and I looked at the figure where they said they tested it empirically, and it doesn't even really clearly show that to me. Yeah, I'm just always wanted to be a bit skeptical. I'm going to stop the share now. I think we don't need to see the comments. But yeah, it was nice to talk about this paper again and get somebody who knows a lot about the nucleus accumbens to point out some interesting parts of it. Thanks, Rudy.
Thank you for your presentation, Stephen. It was good.
Yeah, thanks Stephen for walking us through this paper, and I think there was definitely some interesting findings. Definitely a nice basis for future stuff like this interaction with oxytocin, these ideas of plasticity with MDMA, and kind of elucidating these mechanisms. And thank you, Rudy, and everybody else also for joining or for listening. I think we can conclude soon, unless anybody wants to add something else. Any final comments, questions for Stephen or
Rudy?